Okay, let's dive in. So, what if plants could be more than just, well, plants? Imagine them acting like living computers.
Right, like their genetic code isn't set in stone.
Exactly. It could be reprogrammed, maybe using light or even electricity.
That's the core concept we're exploring today, based on the sources, this idea of a universal biological plant computer.
It feels like a really big step beyond just standard genetic modification, you know?
Oh, absolutely. We're talking about systems that can sense things, actually compute information, and then change their own DNA on command.
Okay, so moving past stabbed traits. You give a plant one thing, and that's it.
Yeah, this is dynamic. It's about changing the DNA in situ, right there, inside the plant itself, where you want it, when you want it.
Precisely. Triggered by specific signals.
And the potential applications, I mean, for you listening, this could be huge, especially in medicine, right?
Definitely. The sources really emphasize that. Think about plants making pharmaceuticals.
Like on demand.
Yeah, exactly. Producing a specific drug, maybe just in one leaf, right when it's needed, instead of a whole factory process.
Or think about diagnostics. A plant could, say, detect a pathogen nearby,
In the air or soil.
or even from a sample you apply, and then it signals that discovery, maybe changes color or triggers some other measurable response.
So living sensors, of course, there are other ideas too, like in agriculture
Yeah.
or cleaning up pollution.
For sure. Optimizing crops, bioremediation. The potential is broad, but the medical side is particularly striking.
Okay, so how on earth do you build something like this? It sounds incredibly complex. What are the building blocks?
Well, it starts inside the cell. You need processing power. That means engineering what we call synthetic gene circuits.
Gene circuits, like biological computer chips, sort of?
Kind of, yeah. You're building logic gates, AND or LON, not U-gates, but using biological parts, DNA, proteins. This lets the cell process inputs, make decisions.
Okay, so the plant cell can think in a basic way, but how do you control it from the outside? You mentioned light and electricity.
Right, that's the external control layer. You have optogenetics, which is using light.
I've heard of that, like controlling neurons with light.
Same principle, but applied here. The sources mention things like opto-CRISPR.
So light-activated gene editing.
Exactly. Turn on a light, make a specific DNA change. Then there's bioelectronics or electrogenetics, using electrical signals.
How does that work in a plant?
One really interesting tech mentioned is organic electronic ion pumps, or OEIPs. Think of them like tiny, precise delivery systems.
Delivering what?
Molecules.
Yeah.
Signaling compounds, maybe the chemicals needed to trigger a change, and they can be controlled electrically to target very specific spots within the plant tissue.
Wow, okay. And then you need the actual tools to edit the DNA itself. That's CRISPR, right?
Yes. Precision genome engineering is crucial. And it's not just the basic CRISPR cutting. The sources talk about the whole toolkit,
Cleaning.
base editors, prime editors, tools that make really specific small changes without a full cut, and also using modified CRISPR systems, sometimes called CRISPR or CRISPR, to just turn genes on or off reversibly without altering the sequence permanently.
So you have the logic, the external triggers, the editing tools.
Yeah.
But the sources also mentioned this big challenge. Spatial control, making it happen in one branch but not another. How do you manage that?
Yeah, that's a key hurdle. It's about integrating all these pieces. The idea isn't necessarily to deliver the entire editing system just to one spot. That's hard.
Okay.
Instead, maybe the editing machinery, like the CRISPR components, is already present more widely in the plant, but it's inactive, dormant.
Ah, I see. Waiting for a signal.
Exactly. And that signal is delivered with high precision, maybe a focused beam of light for the optogenetics part.
Or chemicals delivered right to the spot by those OEIPs you mentioned.
Precisely. That localized trigger activates the machinery only where you want it. Other approaches involve genetically encoding address tags in different tissues or using nanoparticles designed to go to specific cell types.
It sounds incredibly sophisticated. But also, like there's a lot that could go wrong. What are the biggest roadblocks mentioned?
Well, just getting these engineered components into the plant cells efficiently, especially in a mature plant in situ, is still a major technical challenge. Delivery is tough.
Right.
And then there's the biology itself. Plants are complex, dynamic, living things. Ensuring these engineered circuits are robust, predictable, and don't mess anything else up. That's really difficult.
And I imagine there are safety questions too, like what if these traits spread?
Absolutely. Containment, gene flow, biosecurity. These are critical concerns highlighted. Plus, you have public perception and the need for clear regulations. These aren't just technical problems.
Makes sense. So looking ahead, what's the likely path forward? Where does this go?
Near term, probably more sophisticated plant-based sensors. That seems achievable sooner.
Okay.
But the long-term vision, that's the really transformative stuff. Plants as these decentralized diagnostic labs or even mini factories producing medicines on site.
Maybe even receiving digital instructions.
Potentially, yes, that's part of the grand vision.
So wrapping up, this whole concept of a programmable plant computer.
Yeah.
It's incredibly ambitious. It pulls together synthetic biology, electronics, genetics.
It really does. It's a convergence of fields.
But based on the sources, it's not just pure speculation. People are actively working on the pieces. And the potential impact, particularly for you in areas like medicine, seems huge. It could change how we interact with biological systems.
It really could. And it leaves you with a pretty deep question, doesn't it? If we can engineer life to compute, to rewrite its own code on demand, what does that ultimately mean for how we understand life itself and, our relationship with the natural world?
Something to think about.